Welcome. Today we'll analyze a genetics experiment involving peas, specifically looking at the statistical and genetic implications of finding 406 green peas in our sample. Let's visualize our sample. Here we have our 406 green peas. In genetics, we know that pea color is determined by specific genes, with green being recessive and yellow being dominant. In Mendel's genetic studies, he discovered that pea color follows specific inheritance patterns. Each pea plant has two alleles for color. The yellow allele, Y, is dominant over the green allele, Y. For a pea to be green, it must have two recessive alleles. We call this genotype YY. Let's look at all possible genotypes. YY produces yellow peas, YY also produces yellow peas, and only YY produces green peas. When we cross two heterozygous parents, YY, we can use a Punnett square to predict the offspring ratios. This shows us that we expect a 3-to-1 ratio of yellow to green peas. Now let's analyze our sample of 406 green peas statistically. If we're seeing all green peas, this means all plants in our sample must have the YY genotype. This is quite interesting from a statistical perspective. For a plant to be green, YY, both parents must have passed on the recessive allele. The probability of this happening in a random population would be 1/4 or 25%. Finding 406 green peas suggests this wasn't a random sample. This data suggests several possibilities. Either this was a selected sample of only green pea plants, or the plants were specifically bred to maintain the recessive trait. This kind of analysis helps us understand both genetics and experimental design. In conclusion, finding 406 green peas tells us a lot about both the genetics involved and the likely experimental conditions. This demonstrates how statistical analysis can help us understand genetic experiments.